hope that everybody can hear me all right. If that's not the case, just let me know. Okay, so I'll just I guess that you guys had talks already about parasitology and perhaps Cyril touched on Toxoplasma, but I'm just going to remind you of a couple of things about the life cycle of the parasite. So Toxoplasma gondii is a parasite that has its sexual cycle in felids, which are the definitive host. But in the intermediate host, which are virtually any warm-blooded vertebrate, including humans, it's present mainly in two forms. The tachyzoite form, which is a highly multiplicative, highly invasive form, responsible for the symptoms of the acute toxoplasmosis, and the bradyzoite form, which is a persistent resistance form that remains for long term in the intermediate host. So basically, as far as the symptoms go, infection by Toxoplasma is usually benign in immunocompetent competent individuals, because under the influence of the immune system, the tachyzoites will convert into the bradyzoite stage. The key problem is that the immune system cannot get rid of the bradyzoites, right? It cannot be eliminated by the immune system, and it cannot be cured by the drugs that are currently used to treat acute toxoplasmosis. So one key problem is that as I told you, that parasite persists mostly in tissues like the brain and the muscle for a long time, and about 1/3 of the human population, the world population, is infected by the parasite with various degrees of incidence depending on the country. But you can see that it's a fairly high amount of people that basically have a parasite which is present in their brain, right? Which is kind of worrying, although there's no apparent symptoms. So the apicomplexan parasites, and Toxoplasma in particular, are part of the phylum Apicomplexa, whose name derives from the apical complex that you can find in these eukaryotic parasites that contains secretory organelles, for instance, that are secreting virulence factors necessary for invasion and establishment, because these parasites are intracellular obligate parasites. And also they contain an organelle that probably Cyril alluded to before in his presentation about lipid metabolism, the Apicoplast, right? So the Apicoplast has a phylogenetic origin, a complex phylogenetic origin. It's a complex plastid because it originated from a double endosymbiosis. First, a cyanobacterium was phagocytosed to give rise to red alga that was then phagocytosed by an ancestor of the apicomplexan parasites to give rise to that plastid that actually lost its photosynthetic capacity when apicomplexan became parasites. And in some apicomplexan parasites, such as Cryptosporidium, for instance, the plast has been lost altogether. So because of this complex origin, the plastid is surrounded by 4 membranes. And the key message here is that the proteins that are contained by the plastid are of prokaryotic origin, right? So they're largely absent from the host. And I told you that plastid has lost the ability to perform photosynthesis, because that doesn't make any sense, being a parasite in intracellular, or a parasite in a host, and with no more access to the sun, for sure. But these plastids retain 4 main metabolic pathways, which are the Fas2 system for fatty acid synthesis. Again, I'm sure that Cyril told you about that. Heme synthesis, that the plastid is that pathway is shared between the plastid and the cytoplasm and the mitochondrion. Isoprenoid synthesis and iron sulfur cluster synthesis. So the second message here is that not only these pathways are of prokaryotic origin, but they are essential, or at least very important, for parasite fitness. So altogether, that makes it that makes the plastid a very interesting drug target. And actually, it's already a validated drug target because some medications are targeting the plastid, not as a single therapy, but usually as a combinatorial therapy. So clindamycin, for instance, is being used in combination to treat apicomplex and related diseases. Now, when I told you that the Apicoplast is very important and potentially essential, and in Toxoplasma, the data comes from work which has been performed on tachyzoites, so the parasites responsible for the acute stage of infection. The fact is, there was so far no data concerning bradyzoites, right? And as I told you, because bradyzoites are at the moment incurable, we felt that it was important to address that question. That very simple question: is the Apicoplast a relevant drug target for the bradyzoite stage? So it's a very simple question, but it was actually quite difficult to answer. The reason is, first of all, it's difficult to work with bradyzoites because these developmental stages are not easy to obtain in vitro. There are ways of obtaining bradyzoites in vitro. You can, for instance, apply some stresses such as differences in pH, heat shock, or specific inhibitors of the mitochondrial electron transport chain, for instance, but they're not real bona fide bradyzoites. The real canonical bradyzoites you can only obtain by infecting intermediate host. Usually it's the mouse model that we're using, and recovering bradyzoites from the brains of the infected mice. Now, if you want to modulate protein expression or do anything with these bradyzoites you generate, the problem is that because you're working with a parasite that is located in the brain, of course, you have the problem of the blood-brain barrier, which is actually permeable to molecules above 400 and 500 Daltons, right? So unfortunately, most of the systems we have to study the function of genes in Toxoplasma are basically the same that are available for other apicoplexan parasites, such as Plasmodium. The conditional systems for generating knockdowns, such as the TETOF system, or knockout, such as the Crelox system for excising genes, or the Digrand systems for altering protein expression post-translation, they're all relying on the use of ligands, right? And most of these ligands actually have a mass that would prevent the use, or at least make the use not optimal when wanting to regulate gene expression for a parasite located in the brain, again. Perhaps with the exception of the auxin system, because the IAA that is being used is actually quite small. But not only size is important, but also hydrophobicity and the number of other parameters. So it's not ideal to use the classical canonical conditional systems because you don't really know how much you're going to be able to control the parasites in the brain. So instead, to generate conditional mutants of the Apicoplast, we went for a strategy that would take advantage of the very biology of these developmental stages, because although tachyzoites and bradyzoites are not very different in their global cellular architecture, I should say, apart from a number of organelles which are different, they are known to express a different set of proteins, such as stage-specific enzymes, for instance. So it's very well illustrated here from that transcriptomic analysis, where you have on the first column tachyzoites and then different bradyzoites that have been differentiated with several days after differentiation. And you can see that you really find set of genes that have transcripts which are low in tachyzoites and high in bradyzoites, and on the other way around, transcripts which are high in tachyzoites and low in bradyzoites. So we wanted to take advantage of that to generate stage-specific promoters to turn on or off gene expression. So first of all, we chose a candidate which is called APT1, which is a roughly a phosphate transporter of the organelle, which is important for transporting precursor molecules for both the fatty acid synthesis pathway and the isoprenoid synthesis pathway. So when you interfere with APT1, at least in tachyzoites, you're pretty sure that you're going to mess up heavily with at least 2 of the 4 main important pathways of the plastid. So we use CRISPR-Cas9 to recombine at the APT1 locus and replace the endogenous gene with a similar gene tagged with the GFP and under the dependence of the SAG1 promoter, which is SAG1 is a surface antigen which is specific of tachyzoite. So the idea is that when these transgenic parasites would differentiate into bradyzoites, they would lose APT1 expression. And that's actually what we observed. So these are in vitro differentiation, which I told you are not perfect, but they allow us to visualize the lack of expression of the GFP-fused APT1 after in vitro differentiation. And these are bona fide bradyzoites, in vitro generated bradyzoites, because they express bradyzoite-specific antigens. And we can see here that the parasites not fully differentiated, still expressing SAG1 in magenta, they're still expressing APT1, while the fully differentiated ones that are displaying that marker of the cyst wall, because these bradyzoites, as they mature, they become encysted, so they're present in cysts. They're not expressing APT1 anymore, and that's quite obvious on that one as well, where you see a highly differentiated, highly mature cyst that barely express APT1 anymore, while the non-differentiated vacuole still expresses APT1. So the regulation seems to be working pretty well. And the consequences on the Apicoplast was not immediate, but what we could see is that by using antibodies against Apicoplast markers, we tried a couple, actually not a single one, but we could see that in cysts that have lost APT1 expression, there was a subsequent loss of Apicoplast signal. So there seemed to be an impact really on Apicoplast biology. And on top of that, in these in vitro differentiated parasites, we measured cyst size, and we could see that the cysts were generally smaller than the control cell line. So what we did is that we used, we took advantage of the fact that these the protein that we're expressing was fused with GFP to differentiate bradyzoites in vitro and to isolate GFP negative parasites, so the ones that we know have lost APT1 expression. And we sorted them, and we conducted plaque assays that allow us to assess for growth of the parasites, and for us is a proxy for viability of the bradyzoites. So what we could see is that when we sort out the parasites that are GFP negative, they basically generate very little plaques and very few plaques, okay? So they seem to be essentially non-fit, non-viable. Now, I told you the real deal, the real bradyzoites are the ones that are generated in vivo, not in vitro. So we took that cell line and we put it in mice. And to our surprise, we noticed that actually the many mice actually died in the acute phase of the infection. And we could see by monitoring mouse weight that the acute infection was actually more important than with the control, with the acute weight loss of the mice, and many of them, I would say 80% of the mice died unexpectedly, because we're using a strain. The background strain is supposed to be a model for chronic toxoplasmosis, not acute toxoplasmosis. And perhaps related to that, we actually could show that in the few mice that survived, many cysts could be recovered from these mice. So these mice that survived, that were infected with the SAG1 APT1 cell line, actually generated more cysts, which was not really expected, I should say. But in fact, when we recovered these cysts and we did some imaging, compared with the control that displays a high number of apicoplasts, here we could see that even the large ones had virtually lost most of the apicoplast signal. Okay, we quantified that with a couple of different apicoplast markers. And when we use this time a cytometer allowing to do spectral cytometry, really to eliminate autofluorescence, what we could see is that we in the in vivo generated bradyzoites, basically there's no more GFP expression. It's really tightly regulated, even more than when we did the in vitro differentiation. So that could show us that APT1 is really not expressed anymore in the cysts. And only a few of the cysts express some apicoplast markers. So we really have a tight regulation. And when we assessed for the cyst size, again, like we did in vitro, we observed that these cysts were generally smaller than the parental cell line. And again, we used these parasites recovered from this cyst to do plaque assays. And again, virtually they generated no plaques, telling us that these parasites present in the cyst that we recovered in vivo are not viable. And another interesting experiment that we did is that these parasites that we recovered from this cyst displaying some kind of apicoplast signal to some degree, we recovered them by pepsin digestion of the cyst, and we performed whole cell invasion. And then we assessed for the expression of GFP that we found was largely absent. So APT1 is really not expressed anymore, like our FACS data was telling us. But interestingly, the only parasites that we could find that invaded were still expressing an apicoplast signal. So there were very few of them, but the ones that basically you recover from these big cysts are the ones that still express the apicoplast signal. None of them were found to have lost the apicoplast signal. That tells us that the ones that have lost the apicoplast signal, that are really impacted for apicoplast, are not viable anymore. Okay, so that was a first mutant that we generated that tells us that that basically the apicoplast is probably important, essential. There are good chances that it's essential for bradyzoites, but we generated a metabolic mutant. We wanted to generate another type of mutant, a structural mutant, but it's because it's known in apicomplexan parasites that metabolic mutants and structural mutants of the apicoplasma have different outcomes as far as phenotypes go. So we chose a protein which is called DAPA, which is a dynamine involved in the cleavage of the apicoplast, the fission of the apicoplast after division for proper segregation in the daughter cells during division. And the interesting thing is that when you express a dominant negative mutant with a single amino acid mutation of that protein, you induce in tachyzoites a lack of segregation of the apicoplast. So at the locus that is not essential for bradyzoites, we expressed that dominant negative copy of the gene, this time under the dependence of a bradyzoite-specific promoter. And we could see that it was finally regulated again. In vitro generated bradyzoites express the protein only. And we can see that in parasites, bradyzoites generated in vitro, only the ones that are expressing apicoplast, the apicoplast marker P21, which is actually the protein the promoter was chosen from, are the ones lacking the apicoplast or displaying a signal, a normal signal for the apicoplast. And we actually quantify that. And as you can see here on these striking images, in vitro differentiated bradyzoites, you see a very reticulated apicoplast signal. In these, typically in these cyst-containing parasites, half of them would have a reticulated apicoplast and half of them would have lost the apicoplast. We quantify them. But this time, the apicoplast size was not affected, contrary to the APT1 mutant for the in vitro generated bradyzoites. So again, we went for the real deal. We injected these cell line into mice. And this time there was no acute, no pronounced acute phase of infection, no particular death of the mice. We could see that there was an acute phase because there was weight loss of the mice. So acute phase was supposedly occurring normally. And we could also check that the fitness of the tachyzoites in vitro is not affected with the cell line. But when we did monitoring of the parasites during the course of infection in mice, we could detect some parasites in the brain after 12 days, but after 40 days post-infection, there was virtually no more parasites that were being detected in the brain. So there was a clearance of the parasites in the brain. Okay, so again, pointing to an important function for that protein important for apicoplast homeostasis in bradyzoids. So here's a slide for a summary slide for the two mutants we have generated, because you might have noticed that there is a slight discrepancy in the phenotypes that we observed. So for the metabolic mutant that we generated, in vitro and in vivo, we could show that there was a tight regulation of protein expression. They generated, both in vitro and vivo, smaller cysts. Now, the acute infection was higher than with the control, and we could discuss about that in the questions if you have some, but we think we have some hint as to why this happens. But in any case, the parasites that were recovered from this cyst were essentially non-viable. And then for the housekeeping mutants that we generated, in vitro, there was no difference in the cyst size, but was a tight regulation and pronounced phenotype as far as apicoplast morphology goes. And importantly, in mice, these cell line generated non-viable cysts. They were, after 40 days post-infection, no more parasites were detected in the brain. So in both cases, we could see that impacting the apicoplast led to non-viable bradyzoids. So now it raises the question as to which function or functions linked with the apicoplast might be important for bradyzoids. And as I told you, there are 4 different pathways that are known to be important for tachyzoids. So just to give you a picture, an overview, this is not perfect because this is transcriptomic data, and obviously transcriptomic data has limitations. But interestingly, if we look at the expression levels, so this is the fold change between bradyzoids and tachyzoids based on data from the Laura Knoll's lab, gathered from ToxoDB, what you can see is that heme synthesis is essentially downregulated in bradyzoids, interestingly, while DOXP, so the isoprenoid synthesis pathway, and iron sulfur clusters pathways are even upregulated compared with tachyzoids. So that's interesting. That could hint for an importance, particular importance in bradyzoids. And strikingly, when you look at the FAST2 system, the early stages of the pathway seems to be downregulated, while the later stages seem to be upregulated. So again, that needs to be verified, and to because it's not only the transcript expression, but it's the protein expression and the activity of these enzymes which is important. But that could hint for an importance of several of the pathways in the bradyzoid stage. I'll finish with that, trying to keep my time by acknowledging the people in my lab that were involved in that work. So PhD student Cyril Sanchez did most of the work, but that work has been initiated many years ago, actually, by a couple of postdocs, Mayan Nguyen and Aotioti, in the lab. And this work was a collaboration, a very nice collaboration with the lab of Nicolas Blanchard in Toulouse. We did most of the in vivo work. It was funded by the ANR in a French funding organism. And if you're interested for further reading, the paper just got published a couple of days ago in PNAS, and for free version, you can find it on bioRxiv as well. So I thank you for your attention, and I'd be happy to discuss these results with you. Thank you.
Thank you so much, Sebastien, for such an exciting talk, and congratulations for such a tour de force on turning down like the bradyzoid. I know how much of a trouble it is. So I was not aware of the last slide of transcriptomic, which raised super big questions. I will leave the audience maybe start, otherwise I can fire you already with a couple of questions. Yes, Pawan.
Why DRP1 mutant actually there's no infection in brain? We know the mechanism of that gene function?
Did you hear?
You're talking about what, APT1?
The DRPA, sorry.
Yeah, DRPA1, sorry, yeah, DRPA, yeah. What's your question? Can you repeat, please?
Why the DRPA1 mutant or knockout actually doesn't cause a brain infection? What is the actually relationship?
So we don't know, but what we think is so the phenotype that we could observe in vitro is pretty clear. So you get a problem of segregation, okay? So what we believe is that, and it's interesting because you might or you might not know, but Apicoplast mutants can undergo what is called a delayed death, right? Which is actually a phenomenon by which it's not, the mutants are not impacted straight away, but they need to reinvade and subsequently divide to be impacted. And the reason is that for tachyzoites, probably as they divide within the same vacuole, if you have a partial loss of the apicoplast, which is the case in segregation mutants, the parasites that still retain the apicoplast probably can share metabolites, essential metabolites, with the other parasites in the vacuole. Now, we believe that this doesn't happen in bradyzoites, because bradyzoites, the way they divide is quite different. They don't stay connected to each other. When you see how busy a cyst is with hundreds of parasites inside, they don't stay connected by the basal part of the cell body. So what we believe is that, but of course, bradyzoites take longer to divide because they are slow dividers, but they do divide. So that's interesting to see that we see parasites in the brain at 12 days post-infection. But if we wait for 40 days, which is probably the time they need to undergo several rounds of division, we end up with more and more parasites having lost the apicoplast, okay? And being non-viable altogether. So it's a different kinetic than what we observe with the metabolic mutant.
I was about to say, before I pass the hand to Pushkar, doesn't it look like a super extended apicoplast in this DARP-A mutant? And although we don't really know how it divides, it's not the endodiogeny, it's a monoendodiogeny or something. Do we have something like a massive apicoplast that is shared amongst parasites?
Yeah, you're right. It can look like it's actually Plasmodium schizogony, right? with a big reticulated apicoplast. Actually, I didn't put the face picture on that, but actually it's being shared by it's very reticulated, but it seems like it's shared by a couple of parasites. And sometimes it extends after, beyond the cell body, but it goes into the residual body, but it's not like it's being shared by 4 or 5 parasites. Yeah, we didn't do electron microscopy on that and tomography, but that would be cool to do that. But it doesn't look like a single, apicoplast in the whole vacuole, for instance. That's a striking phenotype, yeah, for sure.
All right, I'll stop here. Pushkar, please.
Sebastien, this is Pushkar here, if you remember me.
Hi, Pushkar.
Hi. So just a general question, for my knowledge, and then maybe extrapolate to your studies. So in the brain infection, do parasites have preferences for a cell type, such as neuron versus astrocytes or glia? And if that is, if they do, then would the host matter on the biology of the bradyzoids and maybe extrapolated to the mutants which you're seeing?
Yeah. So that's a very interesting question. Yes, they do have preference for neurons versus astrocytes, for instance. So that's really important because in fact it's the same kind of thing with muscle cells, for instance. It's striking to see that the differentiation of the parasite happens in specific host cells. And these cells are essentially blocked in division. right? So these are thermally differentiated neurons and muscle cells, because the idea is that the parasite wants to remain for a long time without being disturbed. So you don't want to be in a cell that is actively dividing. And we still don't know what are the whole cell determinants that actually trigger differentiation. And that might be linked to metabolism. There are probably some signaling going on. But yes, it's striking. So practically, some people have been using neuron cultures to try and have an in vitro model system to differentiate parasites, for instance. And this is working quite well, but it's not perfect, because when you actually take primary cells, if you take neurons from mouse brain and mice embryo brains, for instance, you always have some astrocytes remaining in the culture, so you have tachyzoites keeping on dividing, and that messes up your whole experiment. So this is a bit tricky, but yeah, that's a good point. There's a tight link between parasite differentiation and the whole cell type, and it's probably linked to metabolism and to sensing of the host status.
Thank you.
It's all about metabolic adaptation.
You always come back to that.
Signaling. It's about signaling.
Well, it
And
works the
signaling,
same for malaria, actually. Yeah. More questions? Yes, Rita, please.
So when you said there are like joined apicoplast, did you look at the centrosome?
We didn't look at the centrosomes. No, you're right. Usually, centrosomes are believed to be driving apicoplast elongation more than segregation. Here we have a segregation mutant, so we can see that for sure. It's obvious that the organelle keeps on elongating. Right? There's no problem of lipid material being generated or of elongation. It's a problem of segregation, because that dynamine is not cleaving the elongated apicoplast properly. But we didn't do any centrosome labeling yet.
It's all okay. All right. I have a burning question. Just a quick one, please.
Okay.
Okay, quick one. I see one of my favorite proteins in there, in a transcriptome ATS1 being up, and I see the FAST2 being down. Now, I can't stop thinking about the parasite using another source of fatty acids internal to integrate it and make the membrane of the future daughter cells, nothing coming out from the outside, nothing coming up from the FAST2, and actually maybe the tag fueling it towards ATS1 and creating the new parasite this way. I'm sorry, that's not a question.
Yeah, I agree with your comment. I mean, it's.
I usually don't like it's more of a comment than a question.
Yeah, no. But that's an important point. It's so it is true that for a long time people have believed that because brachiozoites are enclosed in cysts, and cysts have a thick cell wall, that they were really cut off from the outside environment, so that they were not really properly able to import many nutrients and so on. I don't think it's the case, because there are a couple of papers that show that many metabolites can actually cross the cyst wall and reach the parasites. But again, going back to what Pushkar said, the metabolic status of a neuron is not the same as the one from an epithelial cell or fibroblast, so perhaps there is some specific adaptation, and maybe they rely more on eternal biosynthesis for some of the precursors this time. So I think that I had the same comment as you when I saw the data for the first time. I was like, that's interesting that part of the pathway is down, and essentially the early steps, and the other one is up. So it means that probably they get some precursor from somewhere else. So that's something interesting to find out. I agree.
All right, we'll have this discussion together. I'm sorry to have to be in Delhi to talk more with Seb than when we're in France. No, that's not true, actually. I'll stop here. Thank you so much, Sebastien, for giving us some of your time for this exciting work.
No problem. Thanks for the invitation. Thanks very much.
Long live India and France friendship.